Yeah, thank you. Thank you very much, Miguel, and thank you for the invitation to come. Thank you also to Jan for inviting me. I know he can't be here, but I'm sure he'll be watching later on. It is really a pleasure to be here. It's been already great to be seeing your university, the facilities you have here, and I know that I'll meet some more of you this afternoon, so I'm looking forward to that. So I'm going to be talking about some really, so two stories particularly that a couple of my PhD students have been working on over the last few years, and it is quite new, so I'm very excited to be sharing it with you. And it's particularly exploring this class of biomolecules, the glycosaminoglycans. So I'll talk about them more a little bit later. But just to give you a little bit of a background of myself, Miguel's already given a lovely background, thank you for that, but I did my PhD way back in 2005, actually at Flinders University in Australia. So that was looking at microscale pattern cell substrates. And then I came over to University of Nottingham in 2009 to start a postdoc there, where I was working particularly with Professor Morgan Alexander to develop some high throughput screening approaches. Kind of outlined one here. This has been a lot of work done at the University of Nottingham using high throughput screening approaches to find interesting synthetic polymers that do interesting biological things. I was particularly interested in materials that can prevent bacterial biofilm formation. I am in the School of Pharmacy though, so I dabble in pharmacy, and we've been doing some high throughput drug formulation, miniaturized drug formulation as well, which is some ongoing work involved with that. And then in 2018, I got an academic position, recently promoted to associate professor, which is very nice. And I've been exploring this branch of molecules, these glycosaminoglycans. And certainly my background in biology in terms of biopolymers, I suspect, like most people, focus particularly on DNA and proteins and RNA, and we have this really nice understanding of how you go DNA, RNA, proteins, this great transcription, translation. We can understand the structure of the proteins from the DNA. We can understand the structure of the DNA from the proteins. There's all this beautiful biology that's been worked out. And we talk a lot about them, and then I think in terms of biomaterials and biomaterial engineering, a lot of our work focuses then around those biopolymers, but there's actually this third class of biopolymers which I didn't know that much about, but also I think is quite interesting, quite a lot of interesting biological properties, and it's something that I think is really underutilized and so then has some great potential for biomaterial applications. And so this is glycosaminoglycans. And when you think about the structure of a cell, certainly if you talk about this to a glycobiologist, they'll all talk about, you kind of know there's the cell, there's the cell membrane, and then there's some sort of extracellular matrix, but there's really a really organized layer between the cell membrane and the upper matrix. And a key part of that is glycosaminoglycans. So glycosaminoglycans are long polysaccharide chains, typically attached to different proteins to form proteoglycans. And they form this, this mesh, really like an extended, branched structure that extends away from cells, from this pericellular space. And it's really important at filtering what comes into and goes out of cells. And so it has a number of different biological processes. You'll see it involves a lot in cell migration. It's really important then in embryogenesis and the way that cells can organize themselves within embryogenesis. Also really important generally, I guess, in cell communication, and it's also exploited a lot in a number of different disease states. So the COVID virus uses heparin sulfate. It has a heparin sulfate binding site that it uses to detect when it's getting close to a cell, and that's what allows the virus to dock with cells. It's a heparin sulfate binding motif. So it's involved in a lot of these different processes. But again, I think they're complex molecules, and they're relatively understudied and underutilized. And this is part because I guess they are quite complex. So there's four main types of these glycosaminoglycans. They are defined by their disaccharide structure. And actually, so there's four here. They're kind of divvy up in some ways to 6. So you have chondroitin sulfate and dermatan sulfate. There's some stereochemistry that's different, and so you can kind of split them up. And you also have heparin and heparan sulfate. If you may be very familiar with glycosaminoglycans, you might not be, but you may well have heard of heparin. It's an important pharmaceutic involved as an anticoagulant. Now these sugars, they have these disaccharides, and you can see on these sugars you have these different sites that can or cannot be sulfated. So you can get these different sulfation patterns, and that is all controlled by different enzymes. And that's actually really important because you have these sulfation patterns that can be varied, and that actually gives a lot of variation within these molecules. So you can have sulfated, unsulfated forms. And then you can have these sequences of disaccharides with different sulfation forms, and those different sequences actually can be biologically really important. And that's absolutely true in the case of heparin, and its antithrombin binding ability is based upon a very specific sequence. And I've just said it's a very important sequence, but actually the way that these sulfation occurs is driven by enzymes. There's no template given. So it's, in many respects, it would seem, there's just some sort of collection of enzymes working together to synthesize these sugars. And somehow you end up with these sequences being formed that have really important biological functionality, but it's not template driven. So how does this all happen? And not only that, you get these, no two heparin sulfates in some ways should be the same, right? You have these really long heparin sulfate, and then the enzymes can add sulfates or remove sulfates as they will. But you tend to get, reproducibly formation of heparin sulfates that has, certain structural motifs that are actually really important for their biological function. So unlike the DNA, protein, RNA system where we can really understand the sequences and understand the functionality based upon the sequence and they all relate together, I think the way that the body goes about producing GAGs is still a bit of a mystery, and then their biological function, although they're implicated in many of these biological functions, it's not perhaps so well understood. And so this has become very interesting for me for a number of reasons. I think probably the primary one is that I think that because of their involvement in biological function, because of their involvement in a number of different disease states, there's some interesting biochemistry here that we could perhaps use for different biomaterial applications. And so when I initially started looking at these, because they are complex molecules, I was interested in first trying to develop some analytical approaches that could be used to study these molecules, particularly within biological environments. And I particularly have worked on a technique called, I've just put SIMS, so secondary ion mass spectrometry. It's a mass spectrometry approach. It's surface sensitive. I'll talk about that a little bit more a little bit later. But what I did find, so I talked about there being 4 classes, and then maybe 6, and I've split them into the 6 different classes here, and I've taken a whole bunch of them, done lots of different measurements. And then I've done some PCA analysis and a whole bunch of analysis behind this slide. But hopefully you can see there's 6 different colors representing the 6 different GAG types. And I'm not going to explain particularly PCA in much detail. Hopefully you can see that they're clustering together and that they're separated, right? And that's significant because it means that this technique is able to distinguish between these different sugars, even though in the case of the dermatan sulfate and the chondroitin sulfate, it's really just looking at some stereochemistry changes. So we have a technique that is able to distinguish the different chemistries, so it's sensitive to the different chemistries present in these sugars. And I'll show you here the PCA model behind that. There's a bunch of different ions that we're looking at. So we're looking at a number of different ions collectively, gives us the information to distinguish these different sugars. And likely these ions are associated with the different chemistries that are associated, and the connection is not necessarily clear to us at the moment. It's kind of a chemometric approach. We've got lots of chemical information. We don't necessarily know what it means, but we do know that it can distinguish the difference between the GAGs. So we have a technique that's, I think, useful for analyzing these GAGs. This is what we got to, and we're using this for this. This is particularly focused, I guess, on purified GAGs. I'm looking at glycosaminoglycans for pharmaceutical applications, particularly, I guess, looking at heparins and being able to better characterize heparins, looking at heparins from different animal sources, and that's side part that I'm not going to talk about too much today. But once we'd done this, we thought, well, can we use this to explore glycosaminoglycans in a more, within tissue, within biological samples? And this is where I guess the start of the first PhD project begins. So I started to work with some, particularly Kathy Murray in the middle is a matrix biologist, amongst other things, stem cell biologist as well. And then in the orange is Lorna Milne, who's a PhD student who's been working with me over the last few years. And I'll talk you through her project particularly now. So we were taking our SIMS methodology. So I show you this here now. So secondary ion mass spectrometry. That means we are irradiating our surface with a primary ion source. This produces chemical fragments that then goes away to a mass analyzer. It is surface sensitive, and we can raster this beam to give us spatial information. All right, so I think that's the key things. Surface sensitive, and we get spatial mass spectrometry, which is, I think, perhaps the unique property. And the spatial resolution of this can go down to submicron. So it's on an information scale relevant to single cells. And at the University of Nottingham, this technique is typically used with a TOF mass analyzer, a time of flight mass analyzer. We've also got an Orbitrap mass analyzer, which gives us better mass resolution. So we have these peaks coming off, which we can define a mass with a high resolution. That gives us much more confidence in the fragments that we're producing. So we're using this technique. We have a set of standards to compare with. And we now also want to start analyzing our cell samples. For a number of technical reasons, it's easier to do imaging with the time of flight mass analyzer. So we've got our cells, and what we want to do is look at two types of cells. We wanted to look at some wild type cells, and then we needed to engineer some cell lines that had knockouts. So it's cells that didn't produce any GAGs or didn't produce any specific types of GAGs. I'll talk about that more in a little bit as well. And we wanted to find ions that were present in our wild type cells, ions that were present with our GAG reference library, but that were absent in our knockout. So this is the ideal. This is us just trying to find ions. We don't really care too much about where they're actually derived from, what's the chemistry, what's the GAG structure associated with them. At this point, we're just trying to identify ions just to see, can we see what's what. So we did a whole bunch of optimization, going through GAG reference samples, and then the idea is that we can then produce a library of ions which we know are associated with GAGs, and then apply them to some tissue samples, ultimately explore some different disease states. And so this is where we're headed. And I'll show you some GAG biosynthesis. So we have our protein here. So we have, these are, for the most part, hyaluronic acid is an exception. All the other GAGs are proteoglycans, they're attached to a protein. So we have our protein here, and then there's this linker region, which all GAGs then have, and then it branches at this point, and you can go off, in this case, form heparin sulfate, or chondroitin sulfate, or dermatan sulfate. And so we engineered some different cell lines. Actually, I didn't, our collaborators did. So we could knock out this particular enzyme here, and then this prevents the extension to any other GAG. So we get this little linker region produced, but none of the actual GAG. So this is our no GAG sample. And then we can knock out these enzymes here. These are the ones that extend heparan sulfate. So we can knock them out, and then we can knock this one out. This is the one that extends the CS and DS. So we developed these three different knockouts, and so we now have these cell. Yeah, please.
So the two arrows, does that mean two options, or is it a branching in the chain?
No. So it's two options. It'll do one or the other.
It's not a branch.
Yeah. No, there's no branch. You would either get pure HS or either pure CS. Yeah. So we have these three knockouts, and so we're looking for. We can now have. In the case of this particular cell line, we can find ions that are generally GAG associated. If we have this knockout, we can find ions that are generally HS associated. And if we have this knockout, we can have ones that are generally CS associated. And so we grew these cells, and we could get images of them. So this is, I guess, the ion image doesn't look particularly pretty. It's probably not what you're used to seeing if you're staring down a microscope. But we can do some various checks to make sure that these particular regions are indeed the cells. And so you can see we've done a region of interest just to pull out the spectra that are associated with the cells. And so we have, so I've just focused on the HS here. I'll show you the CS ones in the next slide, but there's quite a lot, so we'll go through the slide. So we have here, these are the ions that we used for that first cell line. So the one where we had those generalized GAG knockouts, so it stops at the branch point. So we have no CS or DS. So we took those ions, and we could produce then an ion image of those. And you can see actually, we're still producing GAGs in both so this is our wild type, this is our EXT2 knockout, so there's no HS in the sample. So we get still a uniform distribution. This is kind of what we'd expect, right? We expect these cells to be producing their glycosaminoglycans. It's associated with their glycocalyx. We have an even distribution of them, and generally speaking, across multiple biological replicates, we don't see any change. If we just quantify all of those ions, we don't see any statistical significant change. Maybe there is a small drop if we did enough replicates. I mean, I think actually there was some small significant drop, but it's not a very big drop. But when we take our ions, and I should note, right, this looks quite convincing, right? It goes from a signal here, these are our HS ions, to nothing. But of course, we've selected these ions by the fact that they've disappeared, so it should indeed give us a blank image. We go from something to nothing with our knockouts, right? And this is because these are the ions that we believe are our HS material, right? Then there's a number of ions that we see that are just reduced in their signal, and we can see those here, or we get a reduction. So we have these ions that seem to be entirely discriminating for HS, and the ions that are reduced, but maybe there's some overlap with some other GAGs or other species. There's also other glycans present that these could be coming from. Okay, so then just to show, so these are the summed ion images. We can take out individual ions, so I'll show you the mass values here. Individual ions, just to look at their distribution. And the images are perhaps not so clear because there's less signal, but what was interesting, I think, particularly with this particular ion, is that with the knockout, all of a sudden we seem to get these low and high regions for this particular ion. So it seems to be that not only is, although we didn't see an overall reduction in the amount of, a very small reduction, it wasn't particularly big reduction in the amount of GAG. It seems like the cells seem to be compositionally arranging their GAG a little bit differently. Okay, so that's sort of interesting. We didn't see this sort of clustering in the wild type. And you can see the individual ions, what it looks like, for ions on the wild type and then after the knockout. So we did the same sort of thing, simplified it for you just to the summed images now for our CS. The CS knockouts don't actually grow very well, so we get much less cell density. But again, we can find ions associated with CS. We are validating it by the same approach of, with the knockout, we can see complete removal within the wild type. We don't get knockouts, so they're still present. So hopefully that's clear, and hopefully that is already at least convincing, at least in some degree, that we are finding ions that are associated with CS and HS. And we think we could relate these back to GAG structure, but that's a complex puzzle that we're still working on. But what we did start to do is, another sanity check is to say, okay, can we apply them to each other, right? So the HS knockout, we've knocked them out, but the CS should still be there. So we check just to make sure those CS ions are still there and vice versa, right? We'd expect with the CS knockout, the HS ions to still be there. So we did this. We quantified them. So we have our CS knockout up the top. So this is the wild type, and this is just looking at the HS ions. And what we see in the CS knockout is indeed we still get, there's still plenty of HS signal present. So we actually did this in 2 different cell lines just to make sure. So there's 2 different cell knockouts. We do see a bit of an increase here. It's nearly statistically significant. Maybe we need to see some more replicates there, which is interesting. So again, we did the HS knockout. So we have 3 different cell lines where we've done HS knockouts just to check to see whether the CS is still present. Can we still detect that? and indeed in these two cell lines we can. And what was interesting with this particular cell is that we do see an increase in CS. So we've knocked out the HS, and rather than the CS level staying the same, it appears that the CS is actually increasing. Right? And so this is quite interesting. Certainly when I start to talk to the matrix biologists about this, there's this idea that the cells actually, there's these 4 types, maybe 6 different types of glycosaminoglycans, and actually maybe they work a little bit interchangeably. So you do have these proteoglycans which can have, so we talked about the fact that from one linker you wouldn't get any branching, but you do get proteoglycans that have both CS and HS on them. And it seems to us here, this gives a hint that maybe there is this sort of compensation mechanism that if you remove HS, actually the cells can compensate for that some ways by upregulating their amount of CS that they're producing. And we're not the only ones to show this, but this is a nice confirmation of this, and we also support this by doing some other, extracting the GAG and doing some other disaccharide analysis on this. And so we were able to confirm this, which is a nice result, but we could look at it a little bit more deep because this is kind of buried. We've just summed all of the signal together, and it's there's some more details in there that we could look at. And when we did that, so these are different ions associated with the CS. These are HS ions. And when we go for we compare them to the wild type and the EXT, the knockout, some of these ions stay the same, and some of them go up. So it's not like the CS that's being produced by these cells is just more of the same. It seems to be structurally different to the CS that was produced just by the wild type. Right? So not only are we getting maybe an increase of CS, but the CS seems to be modified. And actually, when we looked at these ions in a little bit more detail, we noticed that the ones that were all increasing, when we looked at trying to assign them, all seem to contain nitrogen. And so we could say that these are particularly, the change that's happening with CS seems to be particularly associated with an increase in sulfated N-acetylgalactosamine. All right? So that's one of the two sugars in the disaccharide. We seem to be seeing an increase particularly in that particular sugar. And so that's quite nice. And so it's quite nice to be able to have this analytical approach and to see that change biology. Okay, so I'll have a slightly change. So we thought, okay, we've found these particular ions now. We've been able to, I think, validate reasonably well to say that they're at least CS and HS associated. We don't perhaps fully understand how they link back to the CS structure, but at least gives us a way of exploring CS and DS within tissue samples. And so we started to apply it to some different tissue samples. So start talking about Alport syndrome. So Alport syndrome is a fairly rare disease. It's associated with a mutation to KL4 alpha 5, which is associated with the synthesis of collagen 4 A5, alpha 5 of collagen 4 variety. And it means that these patients don't have any collagen 4, and there's no trimerization of collagen 4. So this is the normal case. This is the Alport syndrome case. This disease is associated with a number of different physiologies, particularly problems with eyesight and kidney function. And the kidney function is what we're particularly interested in because that's kidney function with that filtration, the glomerulus filtration, is associated with a glycocalyx. Anyway, the filtration actually through the kidneys generally is associated with glycocalyx. And so we're interested in, okay, within this particular disease, we know that there's breakdown in collagen. There's a breakdown then within the basement membrane that's associated with the different cells that are associated with filtration within the kidneys. But how is the glycocalyx changing, and how are the different glycosaminoglycans evolve with that changing? And I guess another important observation is with a patient. So if you're pregnant, generally speaking, there's extra strain on your kidneys and you get an increase in proteinuria during pregnancy. So what happens in Alport syndrome is instead of being just like 2 times increase in proteinuria, you get a 10 times increase in proteinuria. So patients with Alport syndrome can go under massive kidney failure during pregnancy. And so you'll see patients going under dialysis for Alport syndrome just during pregnancy, and then after pregnancy, it recovers again. So there's this sort of interesting case of looking at Alport syndrome during pregnancy as to what's going on with the the glycocalyx, particularly associated with kidneys. And so so there's another group that was studying this. So this is looking at the glomerulus. So it's looking at the glomerulus from top down. I don't know how your kidney physiology is. The glomerulus has this Bowman's capsule, which is, I guess, kind of this dark space around here, and you have all these blood vessels, and the membrane of them is lined with the glycocalyx, and that's really important in the glomerulus filtration. So there's a lot of glycocalyx. There's a lot of GAGs associated with this particular region. So we're looking down, and these people were studying. There's a stain just to look generally at GAG, and they were observing during Alport syndrome this general thickening of the glycocalyx. So they could measure that the glycocalyx around these blood vessels is thickening as the Alport syndrome progresses, as the age progresses. I think this is within that mouse model. So this gave us reason to explore the glycocalyx within kidneys. And so this is a mouse kidney. So we're able to image this is cross-section of a kidney. You can see it's, 6 by 6 millimeter, 6 by 4 millimeters, sorry. So this is an entire region. We can scan the whole thing with TOF, but we can also zoom in, and I can't do it, but Lorna's got quite good at being able to spot these, the glomerula. So we can see, zoom in on particular glomerula. And so then we can get a signal of the glycocalyx just from these regions. And so we have images here, and we have a different number of different states. There's a mouse wild type Alport syndrome. We have a pregnant wild type and a pregnant Alport syndrome. And then this is just a human one, just for comparison. You can see the size of the glomerulus change from a mouse to a human. So we have these images, and we can now analyze and extract the spectra of those ions associated with the different GAGs. So we've got all the different types: wild type, Alport syndrome, wild type pregnant, Alport syndrome, human. So we look at all the GAGs. Actually, we generally speaking didn't observe big changes in the amount of GAG with our samples. When we looked at the HS, again, we didn't really observe any notable changes. When we observed CS, we saw this interesting spike in the wild type pregnant mouse. So it's like, the CS, the only change we observed was in wild type during pregnancy. And I talked about the fact that during pregnancy there's extra strain on the kidneys, and it seems, we're suggesting here that one of the ways that the body compensates for that is upregulation of CS to aid in the filtration. And I guess notably, we don't observe that same upregulation in Alport syndrome. And so we can kind of speculate, hypothesize that there's this a two-hit challenge. So in the healthy pregnant case, you have nice organized basement membrane containing collagen 4. And then during pregnancy, to compensate for the proteinuria, we get an upregulation in chondroitin sulfate as well. This aids in the filtration. In the case of Alport syndrome, they already don't have that collagen layer. The basement membrane's disrupted, and there's no opportunity for it to upregulate its chondroitin sulfate during pregnancy. And so when the person goes pregnant, that increased amount of blood to filter just means that it becomes more leaky, and we get an increase in proteinuria. So this is nice. We've been able to observe this. Again, these are summed signals that I was looking at, and again, there's a lot of detail within that summed signal. So we looked at not only wild type, we've also looked at diabetes as well. I've already talked about Alport syndrome. What we did observe, in fact, with diabetes, maybe there's an increase in GAG, but we saw a decrease in the heparin sulfate, which was interesting. And again, when we look at the individual ions, we see that some ions didn't seem to change much at all, and other ions did. And I guess it's still quite preliminary, but we can speculate, at least in the diabetes case, that we're getting a reduction in HS, but we're also seeing a reduction or a change in the heparin sulfate structure. So the sulfation patterns seem to be altered within the diabetic state. what that means physiologically, we're still, I guess, working out, piecing it together. But this is really a new observation that we're able to observe, these changes in these GAG structure in the disease states. And so with that, I'm going to kind of leave that story. Hopefully convinced you that using our technique, we can at least observe GAGs, and hopefully got you a little bit interested in these glycosaminoglycans are intriguing molecules involved in different disease states, and there's some interesting things going on that are quite complex, perhaps difficult to understand, but I think probably are biologically important. And that, I think, leads nicely into, so I was working with Amir, who's an immunologist, and then Charan here is another PhD student. We just here successfully escaped a room together with some of my other students as well, so we're particularly pleased about that. So with Charan, we wondered, okay, we've got these GAGs look to be interesting. The structure seems to vary within disease states. They're biologically somehow important. Can we somehow harness this for biomaterial applications? And so the idea here is to apply these to looking at immune response. And immune response, the foreign body response, is a really complex, multicellular process. It goes through a pro-inflammatory and anti-inflammatory stage. You get recruitment of different cells at different stages. And for healthy healing, you need to go through this complete cycle. And in many cases, that doesn't happen. And you can end up with, in the case of biomaterials, fibroblasts and fibrosis being a really common problem associated with this. And so it would be really nice to be able to instruct this process. And I guess macrophages particularly are a key part of that. And there's two types of understood phenotypes with macrophages. We have these M1 and M2 classically defined. So a pro-inflammatory stage, and there's a number of different cell markers associated with that pro-inflammatory phenotype, and a number of different cytokines that are released associated with pro-inflammatory, and we can also see this anti-inflammatory phenotype. And again, there's a number of different cell markers and cytokines associated with that. And I think it's well understood now that it's not just M1 or M2. There seems to be a spectrum, and the cells can kind of be across that spectrum. So we have this M1, M2, pro-inflammatory, anti-inflammatory, and during the healing process, you need to go from initial pro-inflammatory stage where they can recruit cells, remove any foreign objects, any foreign bodies, and then an M2 phase, which seems to aid within the healing. And when I've thought about this, and I guess going from my high throughput background and large data, and I guess thinking about the fact that these cells, I talked to the biologists trying to design these experiments. What should we be looking for if we're going to find a material that's going to aid healing? And it's a very complex question, right? I talked to some people. You need to have some sort of a material that changes dynamically with the healing process. So at the start, it needs to promote M1, and then it needs to be able to switch to M2. So you need some sort of dynamic, and you can't just kind of go for a M2 healing phenotype. But I'm like, well, which cell markers should we look for? Which cytokines are present? And can this be instructed by just putting particular cytokines in? And certainly people have been trying to do that, and there's a cocktail that's needed. But one of the things that I guess has struck me is that there's always an assortment of different cytokines that are present. And so, why is there so much redundancy? If there's an M1 phenotype, why is there so many cytokines that are produced? Why is there redundancy? And I think probably these producing molecules is always a biological expense. I think the answer probably is that they're not redundant. They're all doing slightly different things. It's a complex process, and they're involved in this. Each one of those cytokines has their own role, and they're interplaying together. And you can't really think about it as just M1 and M2. It's a complex process, and at least that excites me a little bit from a big data, complex data point of view. That there's some complex data analysis that needs to be done. Okay, so our approach, and this goes, I think, back to my high throughput screening sort of background, is, okay, can we take GAGs and can we somehow manipulate this particular process? And so what I did is, I went to the supermarket, the GAG supermarket, and said, all right, let me buy everything. and we're just going to screen everything. And then not only that, we'll mix them together at 2:1 ratios to produce this library of GAGs. And when I talk to the materials biologists, they say that's not a very sensible thing to do. But I just admit that I don't know what I'm doing, so I'm just going to try everything, right? So that's kind of my approach. So we try everything. We absorb the GAGs onto plates. There was some optimization we did to make sure they were still there. Then we take macrophages, which we derive from blood samples. So these are primary monocytes that we then differentiate into macrophages. Then we apply them to our GAGs. We did a bunch of different screens, but I guess that we're particularly focusing on the cytokine screening. So within the plates, we can analyze which cytokines are present after the cells have been exposed to our GAGs for a period of time. And so there was the seven different cytokines that we looked at. Clearly, which GAGs which is difficult to see, and it's, I guess, not important. What we can see is there's a range of responses for our different GAG combinations. And when you look at it, if you just try and stare and compare for a while, which cytokines are behaving the same way and which ones are behaving similarly, there is some overlap. So I think the top row were all the pro-inflammatory and the bottom ones are the anti-inflammatory ones. Some of them do kind of overlap. So I think you can look and see the IL-6, the IL-10. There's some patterns that kind of look a bit similar. I think the TNF-alpha as well. All those three kind of look a bit similar, but there's clearly a lot of variation in there too. And this, I guess, comes back to my question, why is there so much redundancy? Why is it producing so many cytokines? Why not just one, right? And it's because I think there is this different behavior. There's some sort of a complex, it's not just M1 and M2. There's something else going on, right? So anyway, we've got some data processing. We did some cluster analysis just to look at, all right, we've put those cytokines in, we just put them all in together and ask the computer to do, what's similar and what's different. So we have all our different GAG combinations. And so we have our M1 here. Our M2 is right up the other end. So, okay, they were apart. But we observed, this range of behavior. And actually, we could try and cluster them into different sections, and actually it seems like there are these different GAGs that seem to be producing these different sorts of behavior. Right? So then we selected GAGs, we had our select GAGs from those different sorts of clusters, and so that was initially done with just a single donor. You do get a number of different donor-to-donor variations with this. This is, I think, a common problem when you're doing immune response is the donor-to-donor variations, but we then took those, our hit GAGs or our selected GAGs, and then did multiple donors with them, and you can see the different colors, the different donors, and you get some sort of similar responses. There are cases where the responses aren't so reproducible. I think it's the CCL cytokines didn't seem to be so reproducible between the donors, but overall we could see generally some similar response. So we did a cluster analysis now with this data, and I think this became quite interesting, is that now the M1 and M2 are kind of in their own clusters, not so far apart. But we seem to get these other behaviors. We have these two GAGs over here. That seemed to be the response, if you look at the cytokines, is really quite different to either the M1 or the M2 case. So what's going on with that? So again, just trying to make sense of this data, and I'm sure we haven't got there yet. But I did some PCA analysis of this. So we're trying to talk a little bit about PCA. PCA is just trying to produce new variables that are aligned with the main variants within the data set. And the aim of that is to reduce the dimensionality of the data. So we have 7 different cytokines. Can we reduce that to 2 dimensions? Because 7 dimensions is a little bit hard to understand, but 2 dimensions we can put in a graph. And so we did this. And so we did PCA analysis. And we have PC1, which is the main variance. PC2 is the second variance. And what was interesting is that PC2 did kind of seem to be aligned with an M1, M2 spectrum, right? So we And then generally, they're kind of aligned here. They're either going up and down here or across here. We saw variations, M1 and M2 in PC2, and so I've just shown the values of the score in PC2 for the average of those particular samples in PC2, and you can see that M1 is very positive in PC2, M2 is negative in PC2, and then we get some sort of variation that's not particularly strong, mainly associated with M2 response from our different, here we're just showing 5 key GAG combinations. But what's really interesting, I think, is that there's this PC1, the variation in PC1, which was the biggest, and that's just, the maths is telling us that. There's this really big variation in PC1 that's saying that there's this variance within the cytokines that seems to be completely unrelated from the M1, M2 understanding. So these GAGs, for whatever reason, have produced this response in macrophages which really isn't predicted from the M1, M2 understanding and seems to be varying in a way that we wouldn't expect from the M1, M2. So that's really interesting. So we took GAGs 1 and 3, right? So GAGs 1 and 3, they had similar responses in PC2. They had different responses in PC1. So I think GAG 1 is back here, and PC3 was up here, right? So different responses, and I thought, is that somehow biologically relevant? So we put these samples into mice, and this is really preliminary data. I'm showing you, it's very preliminary. We haven't worked out what this means at all yet, and this is perhaps not particularly strong data yet. This is only after 7 days, but we are starting to see, and we've done some histology at this point as well, which I think is probably going to be more convincing, but it's not quite worked up yet for me to present to you. But we are starting to see, so this is a wound closure model. So it's on mice. It's a puncture model. We apply the GAGs to 2 of them. The top one is the wild type. And the images, it's not particularly clear, but over a number of biological replicates, it does look like the wound closure for GAG3 is a little bit faster than GAG1, and both of them are a little bit faster than untreated, which is interesting. At the moment, we're just like, this is just sort of interesting new biology. What's going on? How are these GAGs responding? And clearly, looking at how these GAGs respond in vivo is quite a lot different to our in vitro experiments. And so there's a whole bunch of things we have to work out. But I think maybe it shows a tantalizing hint that maybe there is something different in the way that the cells respond to these GAGs 1 and 3. And I guess hopefully my suggestion to you is that these GAGs have some interesting biological properties and may well induce some really interesting bioinstructive properties, and so therefore worthy of further research. And I think that hopefully is my key take home. So just to summarize, and again, this is the point that I've just made, glycosaminoglycans I think are interesting molecules. I've certainly, as I've been exploring them over the last few years, have become quite interested in them. And secondary ion mass spectrometry has been a really useful technique for us to be able to explore these GAGs, particularly be able to explore that in disease states. We've seen this idea of GAG compensation. So when we remove HS, the cells respond to that by maybe producing more CS, maybe changing the structure of that a little bit. And then I think they have interesting bioinstructive properties, and that we can, in terms of immune response, we're observing, and we certainly don't understand what it means. We're observing this kind of interesting macrophage response, which seems to be different to that sort of typical M1, M2 response. And so that's my conclusions. And finally, some acknowledgements. There's increasingly a number of people that have been working with me on this, which I'm very grateful. Thank you very much to the funders for funding. And then finally, thank you all for your attention.